is Dr. Peri Tobias, a research associate at the School of Life and Environmental Sciences at the University of Sydney. She is a plant biologist and bioinformatician who is broadly interested in the complexity of plant molecular responses to pathogens. She completed her PhD at the University of Sydney after a career in horticulture, arboriculture, and environmental management. And she's been successful now in securing several grants from AgriFutures, the Australian Flora Foundation, and also an ARC linkage grant to investigate myrtle rust. So she's actively collaborating with researchers across Australia, New Zealand, South Africa, and Brazil to try and contain the myrtle rust spread. Thank you, Peri.
Thanks, Jordan, for that introduction. And I just, like to thank you for the opportunity to present at the Society. So we've got some sort of new findings. So it was a really nice timing to get the opportunity to present this data. So the title of the talk is "A Chromosome-Level Genome Sheds New Light on the Mating Strategy for Austropuccinia psidii, which is the causal agent of myrtle rust, which is this pathogen that affects, it's a rust pathogen that infects Myrtaceae plants. And on the left is an image of the rust on a willow myrtle. That's Agonis flexuosa, which is a West Australian plant growing in Sydney, just up the road from me. And up top, a picture from Port Macquarie of the beautiful Eucalyptus grandis in their natural habitat. And these are grown as forestry trees all around the world. And the first Myrtaceae genome was actually this plant. And so the importance of this pathogen is that it affects some of our very iconic trees and plants in Australia. I just also quickly acknowledge some of my collaborators on the genome work. Richard Edwards at the University of New South Wales has been working closely with me, and the mating gene analysis, Jessica and Alistair, and there'll be some further attributions at the end of the slides. Actually, I wanted to get the pointer. So basically what I'll talk about today is I guess many people know what myrtle rust is, but I'm going to go through a bit of a discussion about what myrtle rust is, where it is at the moment globally, and the impacts to Australian plants and beyond. And then I'll just talk about the new phased and scaffolded genome that we've just assembled, and what we know about fungal mating, particularly in the Basidiomycota, and how this new genome helps determine the mating strategy. And then I guess move on to where we can go with this new information. So Austropuccinia psidii is I just have to move, I can't actually see beyond this. Yeah, sorry, moving my screen things around. It's a fungal pathogen. It's biotrophic, requires a living host, and it affects virtually all Myrtaceae plants at the moment. There are a few plants that don't seem to be affected, but it's got a very, very wide host range. It's largely clonally reproducing, and so those pustules that you saw on that first slide showed the yellow spores on the leaf, and each pustule releases huge number of spores which are carried by the wind, and those spores are produced clonally, and so they then spread on the wind, land on a new host, and germinate and puncture the leaf surface, parasitize the leaf, and begin a new cycle of clonal reproduction. The image at the top shows the nucleus, the two haploid nuclei within the spore fluoresced with propidium iodide. And so you can see that they retain their haploid nuclei separately within the spore. So they're dikaryotic. They keep these two separate haploid nuclei, unlike, for example, vertebrates or many animals where Our genomes are all within the single nucleus. These are kept separate. That doesn't work. Just a quick picture, electron scanning microscope image of 6 hours after inoculation with myrtle rust on a Eucalyptus grandis leaf, and you can see the germ tubes. So very rapidly they will germinate and then puncture through to the leaf surface. So myrtle rust the centre of origin for Austropuccinia psidii is South America. You can see the huge genotypic diversity here, there is genotypic diversity here. And it was first described on guava, and it's host specific on guava, the guava rust. So that was back in 1880 or so. And since then, there's been other described Austropuccinia psidii that are host specific within South America on different species. But at some point, it was found that the eucalypts that were planted in South America, in Brazil, were getting infected by this pathogen. So it was believed that there was like a host jump, and suddenly we have this pathogen now infecting a wider range of species. And I guess what we can see on the left here in this graph is that the pathogen then was being found outside of South America and moving out. And then within the last 20 years, there's been a rapid spread globally around the world. So, and those red dots indicate the strain that has been spreading rapidly. And here on the map, we have the strain that is most widely spread globally, and it's the pink one on the map, and that's the pandemic strain, which arrived in Australia in 2010, or was found in Australia in 2010, and in New Zealand in 2017, and the Asia Pacific regions. You can see these dates; they're really relatively recent arrivals. And the other key point about this is this pandemic strain has a very wide host range, as does the South African strain, which is actually different to the pandemic strain, and that was determined in 2013. They found it there. So just a list is present on this website that has the global host list, and it's currently at around 480 known species. This is quite extraordinary for a rust pathogen, which is normally fairly host specific. So the pandemic strain particularly has this extraordinarily broad host range. And you can see this list I've got here, it really only goes down to C, and much longer, of course. Many of these plants are endemic to Australia. Australia is the centre of origin for Myrtaceae, so we have a very wide range of Myrtaceae species. So it was always a worry to think of this pathogen arriving, and now it's here. On the left is a Syzygium jambos, which is highly susceptible to the rust, and you can see how heavily infested it is on the new growth. And what happens is it parasitizes, extracts photosynthates from the plant, and clonally reproduces, or maybe sexually reproduces, and then moves on. And so eventually this kills off a susceptible plant. And you can see on the right there, this little Melaleuca quinquenervia, broadleaf paper bark growing down the end of my street, is repeatedly being infected and is just not really recovering. So that's the kind of end result of these infections, is the tree may die. And in fact, these two very recent papers, 2020 and 21 by Fensham et al., looked at imminent extinctions of Australian Myrtaceae. And I think this statement is quite concerning, that they predict the imminent extinction of 16 rainforest tree species in the wild within a generation, with another 20 species also at risk of extinction. There are 2 species listed that are at risk of extinction that have yet to be taxonomically described. So we're losing species in this country before they've even been described, and that may be the case in our neighbouring countries as well. So we have a pandemic strain that we know of in Australia and in New Zealand. The pandemic strain, as I said, has a very wide host range, but is there a possibility for new virulence to emerge? At the moment, we haven't seen, I guess the broader population, the public haven't seen mass death of our eucalypts in the wild, or, you know, it hasn't made a huge impact because the plants that are becoming extinct or really severely affected are plants that are less well known and understory plants, and maybe not the dominant species in that vegetation community. But if there was new virulence that arrived, that may change. And so what do we have to be concerned about? Exotic incursions. So new incursions, there are, as I showed you, other genotypes in South America. So we need, and there's a South African strain, there may be others. So we need to worry about that. Clonal mutations can occur. The masses of spores that are being produced will be mutating. So there are, much like COVID-19, you know, you will get mutations just naturally occurring. Sexual recombination can lead to new virulence, and somatic hybridization, which is actually the swapping of nuclei through the hyphae. And these are all things that we're unknown at this stage, but at the moment we believe we still have this pandemic strain only. Okay, I'm going to move on to the genome assembly. So the genome, the goals for a genome assembly are this, particularly for a dikaryotic genome. As I showed you, that spore has the two separate nuclei with the DNA inside. The nuclear membrane holds the DNA in separate compartments, so A and B. And what we're trying to do when we do an assembly is we extract the DNA, we sequence the DNA. When it's sequenced, it's still a big jumble of the two genomes. What we'd like to do bioinformatically is then pull these back into their compartments. So get the genome separate so that we can start really understanding what genes or what is present in each of these two separate genomes. Until recently, most genomes have been published as collapsed haploid genomes. So it's kind of a mixture of the two, but collapsed down into one version. So that's been the standard way to present a genome, but we're now moving forward, and really the new developments in the last sort of 4 years have been incredible in the way we can actually start deciphering these genomes into two separate genomes and really start, this is going to really advance biology, I think, dramatically. So the goal then is to end up with your phased, this is called phasing the genomes into the two compartments, and then scaffolding the genomes into their chromosomes, so that you've got the complementary alleles of those genes. So that's the goal. And that has actually been quite hard to achieve until fairly recently. Last year, a big group of us published this Austropuccinia genome, and it was a really big job actually, and it was quite a complicated genome to assemble. But what we found was that it was a very large Rust genome. It's around 1 gigabase pairs, so it's in its haploid state, so very large, and that it was made up of mostly transposable elements, over 90%. So these are repetitive regions that can transpose themselves around. They were formerly called jumping genes. So we see that this is a huge component of this Austropuccinia psidii. But it was also clear, even when we published, that the assembly needed better assembly and scaffolding to determine the chromosome levels, contiguity. So we went back and decided to use the latest sequencing technology to try and fix this. And some people are interested in these kind of details, so I'm just going to go into it a little bit. So we used the same high molecular weight DNA that we originally used from the pandemic isolate that's kept at the Plant Breeding Institute in Sydney. And we sequenced it on the PacBioSequel2 machine at the AGRF at University of Queensland in 2021 at a cost of $7,800, so pretty pricey. That's because of the size of the genome. And we got some pretty nice high quality reads, and the mean base pair length of those reads you can see on this graph is around 15,000 base pairs. That leads to nicer overlapping to build the genome. It was assembled using the HiFiasm software and with 33 times coverage, and we incorporated Hi-C data, which is the chromatin conformation data, to phase the genome. So the raw data is available there. The two genome assemblies that came from that software were around 1 gigabase pair each, so that was really good to have that. And then the genomes were independently scaffolded with this pipeline by Aiden Labs, and there's a manual curation step using a graphic user interface tool called Juicebox, which allows you to kind of actually visualize where these chromatin interactions are happening and see where misassemblies have taken place, and then just kind of correct them manually. So that part took some time, but was quite, it ended up being quite a useful tool to clarify these chromosomes. And what we were able to see, so these boxes, blue boxes, represent the chromosomes. And for the first time, we were able to see that the Myrtle rust has 18 haploid chromosomes. So that was, you can just count them. They just turned out to be very nicely in their spot on this heat map. Now, I suppose some people are kind of surprised that we didn't know how many chromosomes there were already, but visualizing chromosomes in rust has been actually very difficult, mainly because with cytogenetics, you need kind of chromosomes of a decent size to be able to visualize them. And there's been various attempts over time to even count the genome, the chromosomes in smaller rust species that are a very important pathogen of cereals. And it hasn't been very conclusive, but the last couple of years, there's been at least three genomes that have been published that actually have, so I think it's the oat crown rust, the wheat leaf rust, and the stripe rust have all found that there are 18 haploid chromosomes. And that was through this particular method we've just done, which is using Hi-C and phasing the two genomes. So it's actually happened bioinformatically before it's been able to happen visually in the lab using cytogenetics. So it's good to see that our rust, even though it's not in the same broad group as the Puccinia, it actually has the same number of chromosomes. We also assembled the mitochondrial sequence at 94,500 base pairs, and that is included in the genomes that have been loaded to NCBI as well. Some statistics on the genome are here. So if we can focus on the colours here, red is, I've called it HAP1, if we think of it as the one, genome 1 or genome A, and HAP2, genome B. So we can see that if you look at this graph here, you can see the size of the genome and the number of chromosomes, if you will, or contigs. You can see that both our genomes, the HAP1 and HAP2, both almost fully assembled at around the number 18. So it's kind of like at 18 chromosomes, we seem to have assembled just about everything. And then there's a few small pieces that couldn't really be assembled, and these might be highly repetitive, or some of them might be mitochondrial or other bits of DNA that couldn't be scaffolded. So it makes it look like it's pretty clear that we've done a reasonable assembly here. We've got some numbers here, which are the number of sequences in the assembled genomes. And even though the number, this is the version 1 genome that we've already published here and here, even though these numbers for this new version are higher, what we've been able to see is that the previous version had kind of put things together that perhaps shouldn't have been put together. For people who have some interest or knowledge, there's just some statistics on the mean lengths of these regions and the N50 and L50 counts, as well as the GC content of the actual genomes, which has stayed around about the same. In this BUSCO results compiled from BusComp, which does a sort of comparison of the conserved genes, you can see comparing the version 2 genome to the version 1 genome. I guess the main thing to concentrate on is the blue, the pale blue, and to see how many complete genes we've got now as compared to the previous version. And again here, the complete gene complement versus the previous version. So we've got a much better assembled genome. When we look at the synteny of these chromosome scaffolds in our new version of the genome, so in HAP1 and HAP2, or A and B, you can see these very clear synteny blocks, a few strange bits here, but basically we've got matching synteny across a large portion of our 2 haploid genomes. And I guess the key thing to look at on this graph is that we've got the dots represent telomere locations. So these you would expect to see at either end of the chromosomes. And you can see we've got dots here. We've got something strange here with these dots occurring right where the synteny breaks. So it looks like we probably need to adjust some of these scaffolds based on this. But we have some we have telomere to telomere scaffolds here in a lot of cases, which is pretty encouraging. And we compared our genome with, I guess, the most up-to-date and well scaffolded and phased genome around, which was just published in March, March 2022, so this month. And you can see that, and this is the wheat leaf rust genome. You can see that we have these, most of the time we are dealing with comparable synteny blocks, but what you can see is a couple of things. The size of those chromosomes compared to Austropuccinia psidii, it's a much, much smaller genome. So the chromosomes are much smaller. And what you can see is also this fanning out of these syntenic regions. And this kind of matches with what we found with these very large intergenic regions in Myrtle rust that are repetitive or transposable elements. So those genes that are very syntenic with each other on these chromosomes are not, they're very widely spaced in the Myrtle rust. So that's, I think, of interest and something, you know, kind of confirms our previous findings. I just threw this small graph in it below just to show you how the version 1 genome stacks up against version 2. And you can see that there's a lot of things that have moved around that were, mis-scaffolded previously. Now I'm going to launch into a little, I've had a bit of a fast track on learning a bit about fungal mating, but I find it really fascinating. And so the Basidiomycota fungal mating, this is a really great chapter, this fungal sex by Colo et al. And we using this to investigate our genome a bit. So the fungi, Basidiomycota fungal mating, it's based on reciprocal exchange of these diffusible pheromones. And what it requires is heterozygosity at the pheromone receptor in one haploid and the homeodomain transcription factors. So we have to have heterozygosity at both these loci for compatibility in mating. So mating type, the type of mating strategy depends on linkage of these two loci. They're called MAT genes. If we have independent assortment of these alleles, it leads to 4 gamete types, which are tetrapolar. It's called tetrapolar mating strategy, and it means there are fewer potentials for outcrossing. But if you have linkage between these two loci, you end up with 2 gamete types, and that means you have a bipolar mating strategy, which is allows more potential for outcrossing. And I just refer you to a good explanation here of rust mating by Alistair McTaggart. Even though some of the details on myrtle rust were we've found we have new evidence now for, the actual explanation of the mating is very good. So in that chapter that I mentioned previously, there's a phylogeny, and that is the thing to probably make note of here is, so this is published in 2017. So there were, at that point, the rust fungi was very unclear what the mating strategy was, whether it was tetrapolar or bipolar. And although there had been some looking for these particular mating genes, so the HD, the homeodomain transcription factor genes, and the pheromone receptor genes, and they found 2, 2, and 3, and 3. So, but it was very unclear what the mating strategy was, and that's because it's a biotroph, and these sort of recombination tests are very difficult to do. So we went and investigated our new genome and compared it to the genomes that are available for Austropuccinia from South Africa and from Brazil. And what we found was 2 copies, and there are 2 homeodomain genes that are linked, and there are 2 copies, one on HD1 gene on scaffold 3 in one genome, and one on scaffold 3 in the other genome. And we find a similar pattern in the Brazil and the South African strains. So we have and these are very diverse. So there's a lot of variation even within, between these 2 haploid genomes within the same species. And the same for the homeodomain 2 locus. So then when we go and look at the pheromone receptor genes, we found 2 copies of this 2.1 pheromone receptor gene, and 1 each of these. Much as was found in that phylogeny, there were 3 whole genes of these pheromones. We actually have 4 because we've got an extra one here based on the 2 haploid genomes. These are likely to be the results for Brazil and South African genomes are likely to be due to a collapsed genome presentation. So what does this look like on the actual scaffold, so on the chromosomes? What we see in HAP1 or HAP-A is the 2 HD genes are linked on scaffold 3. And we have 2 linked on scaffold 3, and these are different. Okay, so we've got different, so this is heterozygous. We find we have 1 copy on each of the, this is still being resolved, but likely to be scaffold 4 in each of these 2 genomes. So these are not involved in mate compatibility because they're identical. But what we do see is this very interesting thing here. We've got one of these ST 2.3 genes in one genome and a 2.2 gene in the other. And these are actually quite different. They're completely different pheromone receptors. So this is showing that this genome and this genome have different, have compatibility. They're heterozygous. So this is in our single genome for the rust. And that matches what we see in the wheat leaf rust as well, which was the other genome I just presented data with. When we go back to our, this is just sort of cut a close-up, I guess, of the synteny map, and you can see where these genes occur. So we see that on scaffold 3, we've got the homeodomain genes here. We've got these ones are the same, the 2.1s, and then we've got one each of these. And that is what we see again in the wheat leaf rust. So this is actually tetrapolar mating strategy. It's very, very clear. I'm probably labouring this a little bit, but I'm just going to talk about this in a slightly different way. And these images came from Alistair, and I think they're quite useful to understand. But so here we can see this as the nuclear membrane, perhaps, with the genes inside, and haplotype 1 and 2 of our Myrtle rust. And we can see clearly we've got different versions of these pheromone receptors in each of these nuclei, and therefore, and these are different, so therefore they are mate compatible in the single genome. And the way it works is that the products of meiosis, which are basically, you just have one of these in your meiotic, your product. These spores would germinate on a leaf and their hyphae could interact. So if you think about it like this, you know, HD1 little a, little b meets HD1 big A, big B. Those two are compatible. 2.2 and 2.3 are heterozygous. So both these loci indicate compatibility. So these two can mate and produce a new strain. Whereas here we have HD1A, big A, small b, can't mate here, and it can't mate here. And then the question arises is if we have a different strain arrive in Australia, perhaps the South African strain, and it has these loci here at the HD, these genes at the HD locus, and this particular variant of the pheromone receptor, can this mate with this one? It's probably likely that it can. And so we need to be thinking that that is a possibility and that this particular mating strategy will allow interbreeding. So that's concerning. We don't know if that's happening yet in the wild, even within our own pandemic. So where to now? I'm going to just read this. So this is again from this Colo et al. At the population level, polymorphism of the mating type genes is an exceptional aspect of some basidiomycete fungi, where selection under outcrossing for rare intercompatible allelic variants is thought to be responsible for mating types that may number several thousand. Okay, so I think that that statement is alarming. It implies that there may be huge opportunity for recombinational diversity and potential new virulence. So we need to be vigilant. We need to not permit any new incursions into Australia. We need to monitor for changes in populations that are already here. And we now have these useful tools to monitor for incursions and population changes with this new genomic information. So the key findings are that well, we've found that Austropuccinia has 18 haploid chromosomes, and we're now working to try and visualize these chromosomes. My colleague Peng Zhang is working hard at this and getting some success, which would be great. The mitochondrial genome's been assembled and is available. Actually, I should say it will be available. It's still being processed at NCBI. Austropuccinia psidii has tetrapolar mating strategy. So it is possible for it to mate based on these two loci. Based on the pheromone receptor mating genes, it's very likely that populations formerly structured by host in the centre of origin can outcross on shared hosts. And we can use the polyallelic nature of mate genes to monitor new incursions and local population structural changes, and that's what we're actually working towards now with regards biosecurity and just understanding the pathogen. And so that's the end of my talk, and I'm welcome questions and comments and errors I may have made in mating in fungi. But yeah, the funds for the sequencing were generously provided by Australian Flora Foundation, and my other collaborators who will be, and there may be more that contribute further down the track on the genome and the mating genes. So thank you.
Thanks so much, Peri. That was great. And very nice, clear explanation of all the systems. There is one question in the chat from readers. Fantastic talk, thank you. Has anyone observed teliospores, and is there evidence for recombination in the population?
Yes, and yes. Not in our population here, but there's evidence for now, this is where I go a bit hazy because I haven't read Alistair's paper, but they were able to develop show mating cross using basidiospores on a universal host. So that does occur, and I think that the teliospore question it was answered some time back, and yeah, we do see teliospores in Australia. So that is there.
Are the MAT genes the same as those responsible for dolipore scepter and clamp connections in some basidiomycetes? That's from David Guest.
Oh, David.
Yeah, right.
There was something about the clamp connectors that I was reading in that chapter, but I don't think we know, or we may know, but I think with the Puccinia, it's just with the rust, I think it has been really difficult to determine these stages, but I may just, it's not my area of expertise, so I may just not know.
David knows. David, do you know? Is anyone else doing this sort of work with their populations, like all of the different strains, races in other countries? Are they also doing this sort of, high genome mining?
Well, the genome is, like, no one's had access to it yet. This is a brand new genome. So unfortunately, the previous genome was giving us the wrong picture with regards to mating gene structure because of the scaffolding. So it's really only now that we've been able to really kind of dissect these two haplogenomes properly that we can see this mating strategy. In fact, like literally yesterday or the day before, I was preparing this talk and I was using another phased genome to kind of compare against myrtle rust, and it's a wheat leaf rust phased genome that was published last year, and it appears that it actually is incorrect as well. So it's not, these are sort of not, the way it looked in that genome was wrong too. So I do think that these are just new things that we're only really able to find now. So it's a new era really in understanding this.
Cool. Did anyone have any more questions or comments? If not, I'd just like to thank Peri again and thank everyone else for attending. And also with all of those mentions of Alistair, very pertinent. He is next month's speaker with a talk titled Sex and Drugs, unsurprisingly. He will be discussing his magic mushroom work that he's been starting on our native magic mushroom species. So that should be fun. That should be popped up on the website soon for everyone to register for. But thank you all for coming today. And thanks again, Peri. Okay.
Thank you.
Please do the survey when it finishes.